Hey everyone. When it comes to longevity, a big focus of the channel is slowing the rate of aging using the testing, tracking, and intervention-based approach. But this approach only goes so far. What about rejuvenation? Today, doctors Michael Levin and Leo Lopez will present evidence for restoring bioelectric gradients as the potential top dog, or what I perceive to be the potential top dog, for treating aging and cancer and for human rejuvenation. So welcome, Michael and Leo, and let's get into the presentation.
Excellent. Thanks for having us. I'm Mike Levin. This is Leo Pio-Lopez, who's a senior scientist in my group. And what we want to do today is actually talk about three specific things, and one of which is bioelectric. So there are three fundamental theories of aging, which have to do with what is it that's the ultimate root cause of it. There are the damage theories, which say that aging is basically accumulation of the sort of thermodynamic noise and damage, and that paints aging as fundamentally a problem of physics. And then there's the programmatic theories, which suggest that evolution has actually favored this kind of phenotype, a decline during aging. And that basically suggests that aging is due to a problem of biology or evolution. And we are going to talk a little bit about a third kind of theory, which is basically a cognitive theory, which suggests that aging is, at least in important part, a problem of cognition or cybernetics. It's a fundamental thing that doesn't require aspects of physics or biology. And of course, these are not mutually exclusive. So in our lab, we have three current sort of contributions to the aging field. We don't primarily, my group doesn't primarily work on aging, but we have three different things that we're working on, and we're going to show you all of those. One is this notion of a loss of goal directedness as a fundamental cause of aging. Then we're going to talk about the degradation of instructive patterns that happen to be bioelectric and aging as a consequence of the degradation of this instructive information. And then something fairly amazing that Leo discovered called atavistic dissociation. And that is one of the consequences of aging. Okay, so this is and you can find some of this here, but basically the idea that if aging is going to be the kind of a widespread body system wide reduction of function and all of these different kinds of things, we need to ask what is upstream, what is actually being disrupted to cause all of this. And one of the things that we've proposed is that aging is a loss of morphostatic information. It's a loss of information that normally keeps the cells in the body orchestrated towards a healthy, functional structure. And specifically, I want to introduce here the idea of bioelectricity. So it has long been known that bioelectricity is important in the brain. It is the thing that makes you more than a pile of neurons. It's the reason that you know things and you have goals that none of your individual neurons have. And it basically arises because individual cells, such as neurons, have ion channels in their plasma membrane that allow them to generate a voltage gradient, which may or may not propagate to neighboring cells. And that allows the system, as you can see here, this group did this amazing video of a living zebrafish brain, allows the system to do computations and have goal states and preferences and all of these things that individual cells don't have. Well, it turns out that this amazing ability of using electricity as a kind of cognitive glue to bind together subunits into a larger system that knows things and solves problems in a different problem space is extremely ancient. This is not specific to electrophysiology of the brain. This is something that evolution discovered around the time of bacterial biofilms. So every cell in your body has these ion channels. Most cells are connected by gap junctions, these electrical synapses that allow them to form electrical networks. And what you can see here, I'll just play this video. We now have developed tools, molecular level tools, to read and write the electrical information in tissue. So whereas you might read the information in the electrical states of the brain to hopefully do neural decoding and sort of try to understand what the cognitive information content is of this animal, you can do the same with the body. And so this is a voltage-sensitive fluorescent dye used in an early frog embryo. It's a time-lapse video, of course, but what you can see here, there's a lot going on. This is one frame out of that video. We call this the electric face. And this was discovered by Danny Adams in my lab, who found that long before the face becomes regionalized into the eyes, the mouth, the placodes out here, the anterior neuroectoderm carries a bioelectrically encoded map of what the future face is going to look like, right? If you can read out those voltage gradients, which you can see here, you can literally see where the eye is going to form, where the mouth is going to form, and all of those kinds of things. What we found over the years, and we've been at this for quite some time now, is that these standing bioelectrical patterns within the body are encoding large-scale anatomical features. For example, in planaria, so here are these flatworms, and planaria are a very important model system for aging research. We'll talk a little bit about that. But in these planaria, there's a voltage gradient that allows every fragment. So when you cut this thing into pieces, they regenerate. So this middle piece right here normally will make exactly one head, one tail. There's a voltage gradient that says you should have one head and one tail, and that is exactly what they make. We can go in and reprogram that electrical gradient. We can convert it into this pattern, which basically says two heads. And when you take those animals, which still have anatomically, they're normal. Molecular biology is normal. They have the right head markers in the anterior, not in the tail. But when you cut them, they make two-headed animals. Okay? This is not Photoshop or AI. These are real animals. And that's because the set point towards which all of the cells are working when they regenerate the animal has been re-specified to say two heads, right? So control of head number, control of a large-scale body plan is stored electrically. There's nothing genetically wrong with these. We didn't touch the genome. We didn't edit the genome. This is a physiological a layer of information. And then just one other example I'll show you is using the same signals to ask the tissue to grow new organs. So here's a side view of a tadpole. Okay, so a vertebrate animal. Here's the primary right eye. Here's the brain up here, the mouth. This is the gut. And what we've done is inject an ion channel that induces a particular bioelectrical state that causes the cells to receive a build an eye here signal. We're not talking to the stem cells. We're not trying to tell it how to build an eye. We have no idea how to build an eye. What we do know is the high-level prompt, the trigger that when you establish that bioelectrical pattern, the cells interpret it as here's where we need to build an eye, and sure enough, that's what they do. And so this is the idea here. And so, Leo, you want to talk about this part?
Yes. So as Michael introduced it, so if bioelectricity is an interface to high-level anatomical control, we wanted to know what's happening to the biological pattern during aging. So we wrote this review perspective paper in 2024, and basically we crossed the literature about everything about ion channels, gap junctions, and everything about aging. And we found a lot of links. And in this paper, so we made three main hypotheses. So the main one is that aging is due or partly due to the corruption of the biological pattern over time. So here, what we can see is some sort of a corruption. So it's more like it's more blurry during aging. It could be something else. The second hypothesis is that aging is due to the loss of cellular competency, so to interpret the bioelectrical pattern. Or both of them, so in a very bad negative feedback loop, so leading to aging. You can go to the next slide. And part of the answer has been realized in the lab by one of the postdocs, so Hamid Sediki. So he found senescent associated membrane depolarization. So meaning that if during aging we have an accumulation of senescent cells, we should have a corruption of the biological pattern with this senescent associated membrane depolarization. And so this is what we can see also in, I can't show, but in the figures. And we also found very specific biological pattern of immortal hydra compared to mortal hydra. So this is the work of Stefania Kapsetaki, that is also a postdoc in the lab. And so for us now, bioelectricity is definitely so involved in aging. And I think the next slide, yeah, I think it's yes. And now, so this is what we have done for the bioelectricity of aging. And now we are going to go more into the loss of goal-directedness theory. And we are starting by introducing you how we see biological systems. So with this cognitive lens, So the cognitive lens on biological systems is that we see them as composed of a multi-scale competency architecture. So it means that we have cognition at multiple levels of biological complexity. So here we don't have to understand cognition as like high-level cognition or consciousness, but more as cognitive capabilities or cybernetic ones like problem-solving capabilities with memory, learning, anticipation that we can find in a lot of different uh, um, levels. So, like chemical networks, we have neural networks. Cells can learn and memorize. And so we have with this view, we have a scale of cognitive levels that we can see here, like an example of Wiener. And we want to model so biological systems as cognitive. If we by doing so, we have better and improved predictive and control capabilities. And we like to use the cognitive icon to define let's say the sophistication of the agent, the scale of its goals, so goal directedness, and so it represents. We will see that after, but it represents, let's say, the size and shape of the goals that an organism or an agent can reach.
Great.
Yes.
So, so in the first part of the talk, we mentioned these bioelectrical patterns and how they define specific structures within the body. And so I want to now talk a little bit about how it is and that cells interpret these patterns. And I'm going to make the claim that what these patterns represent are biophysical encodings of goal states that the cellular collective tries to implement. And it's very important that we understand the storage and the interpretation and the potential degradation of those goal states, because they are what drives first the development of the embryo and then the upkeep of the body throughout health and adulthood. And in order to start talking about goals in living systems, we have to understand that goals are not just for complex brainy organisms. They're not just for behavior in three-dimensional space. Biology has, as Leo said, this multi-scale architecture where every level of organization, every scale is able to do certain things that are well recognizable to behavioral scientists. For example, it is now seen that even molecular networks, so never mind even cells, but the molecular networks or pathways within cells are capable of several different kinds of learning, including habituation, sensitization, even associative conditioning. So Pavlovian conditioning. So we are, for example, trying to take advantage of that in our lab to train actual pathways for things like the drug conditioning and so on. And so here we come to the part that's really critical for aging. This is a nice diagram that Jeremy Gay drew about the ship of Theseus. So if you remember, the point of the ship of Theseus is that you have this famous structure, the ship, and over time all the planks are damaged and have to be rebuilt. And so the question is, after a certain amount of time, when every single plank has been replaced, is it still the ship of Theseus? And people have used this analogy for the body, right? The materials of the body come and go. The cells senesce and die and are replaced over decades, and yet the same body continues with some modifications. So puberty and then various other changes, right? So there's some maturation. There's some changes, but overall, the point of longevity is to maintain that large scale coherence. It's okay if the cells age and die, but the large scale organism has to go on. And the interesting thing I think about this ship of Theseus analogy is that the ship isn't the physical, but the boat, the wooden boat. That's not the ship. The ship is actually the pattern in the mind of the replacement machinery, which are the workers. In other words, as long as the workers have a correct plan, as long as they understand what the ship of Theseus is, and they all agree, by the way, what it is, they all have the same plan, then what's going to continue is the pattern. The materials will flow in and out. But in that sense, we are a process, not a structure. But that's fine as long as everybody has the same plan for what's going on. And so I want to introduce you to a sort of champion of that process, which is this planarian flatworm that we mentioned a moment ago. So asexual lines of planaria show no signs of aging at the level of the organism. They are, for all intents and purposes, immortal. Individual cells age and die, but they are continuously replaced, regenerated, in fact, in place, and the worm simply goes on. And so now we want to know a couple of important things, right? We want to understand, first of all, how this plan in the body is stored. And we mentioned before that bioelectricity is really important. So partly it's stored bioelectrically. We want to understand, can we rewrite that pattern? So as Leo said, when the pattern becomes blurry or degraded, can we reinforce it, right? And so we've developed techniques to sharpen and to reinforce those bioelectrical patterns. And in particular, we want to understand what is actually happening when those patterns degrade, and how do cells use these electrical networks to align towards common purpose. Because no individual cell knows what a body is or how many eyes you're supposed to have or any of that, but the collective absolutely does, as you know from the regeneration experiments. And so as Leo briefly mentioned, there is this notion of a cognitive light cone, which is basically the size in both space and time of the largest goal states that a system can pursue. So here's a single cell. This is a unicellular organism. And basically, all it cares about is the local environment here. It has tiny little physiological goals, metabolic goals, and so on in a tiny little region of space-time here. But something amazing happens during the evolution of multicellularity and during embryonic development, which is that individual cells come together to be able to pursue enormous, grandiose goals. So here's a salamander limb. You amputate the limb, they grow. Basically a very good quality copy, and then they stop. And that's the most amazing thing about regeneration, is that they actually know when to stop. So they have this region of anatomical space that they try to get to, and then and if you try to deviate them, they try to come back. So this set point, this homeostatic set point, is enormous, and that's because the cells are joined into physiological networks. That, of course, has a failure mode, and that failure mode is known as cancer. And what you were seeing here is some glioblastoma cells running around, because when cells disconnect from this electrical network, they basically roll back to their ancient unicellular self, because they can no longer remember the large pattern that they were working on. They're not more selfish, they just have smaller selves. As far as they're concerned, the rest of the body is just outside environment. But the boundary between them and the outside world has shrunk now to the level of a single cell. This will become important in the story that Leo is going to tell in a minute. So Leo, talk about the loss of goal directness.
Yes. So we wanted to understand aging is this multi-scale competency architecture. And so we developed a model based on neurocellular automata, that is basically a cellular automata on steroids, where we have a grid of cells, and each cell's behavior is controlled by a neural network that is here equivalent to a gene regulatory network. And the collective cellular behavior will give rise to system-level behavior, that is here, in this case, morphogenesis. And the key point here it is that we evolved this neurocellular automata to perform only development and nothing else. So we didn't select organisms for good morphostasis or aging, just to do development, so to reach the appropriate target morphology. And this is what we obtained. So here what you see is the lifetime of different organisms that have to reach. So the target morphology is the smiley face, and what we can observe is that all the organisms reach perfectly the adult morphology, and then we start to see a degradation of the anatomy, and sometimes they even lose organs like the eyes or the mouth. So meaning basically death. So what is very important here? It is that we have aging, so we interpreted it as aging. We have it without any external perturbation or accumulation of damage. It's due to it's a disorder of the collective behavior here. And so we interpreted it as aging as a loss of goal directedness in the anatomical space. So the cells don't have any high level anatomical goal anymore, and after development. And yes. And in this simulation, we also found a very interesting thing, because we could reactivate regenerative capabilities. So on the right, we can see the stars. The stars means that we injected embryonic states in the cells that lost the organ, And what we could observe is that we reactivated developmental programs that are basically regenerative programs. So it's like here we have rejuvenation of the anatomy by this targeted simulation, and it um, and so. And this is related to what we have also right now in partial cellular reprogramming, for example, when they injected like Yamanaka factors that we can find also in the embryo, and that led to regeneration, for example, of the eye or rejuvenation. Yes. So this theory of the loss of goal victiness can also do also a few predictions that never-ending anatomical goals may lead to minimal aging and lower cancer rates. So why? Because continuous growth or whole body regeneration or never-ending anatomical goal would be, there is no end to that. And we have also organisms like that, so like lobsters, axolotl, we have some trees or planaria or Greenland shark that show negligible aging and extreme longevity. And also why for cancer, it's because if you have a continuous anatomical goal-directed net, it's like there is a maintenance of the morphogenetic field, the cellular alignment that will avoid the cells to pursue their own cellular goals. So the going downwards, let's say, in the scale of the goals, like for example cancer.
Yeah. So just to summarize the amazing thing that Leo just told you is that in the simulation there is no underlying damage. We are not modeling any kind of entropic disorder at the lower levels. We do not have any kind of evolution or in this for death. So there is no selection for aging, and yet what we find is that the goal-directed system, which is a collective of cells trying to minimize distance from a specific shape, that smiley face is basically the electric face pre-pattern that we showed you. When they have achieved that goal and they don't have a new one, they basically start to disband, right? They've it's a very deep philosophical question. What does a goal-directed system do after it has met its goal? It has achieved its goal and it has nothing else to do. And so this shows that there's perhaps another previously unknown cause to aging, which has nothing to do with physics or selection. It is fundamentally a cognitive problem, and there are others. So cancer is one of those, right? As we just discussed, there are single cells that have tiny little cognitive lycones, and they can work together in networks to have a much bigger ability to store memories. But we know that can go awry, and in cancer, basically, it's a problem of the scaling of goals, and it doesn't have to be a genetic damage. It can be many other causes. Could cause a physiological dissociation of the network, causing a reduction of the scope of the size of set points that these cells can pursue. So what about aging in cells?
So we wanted to check if we had this some sort of an atavistic genetic expression during aging, like in cancer cells, and to test it. So what we did is like metaphylostratigraphic analysis of a lot of different cells and tissue transcriptomic data during aging. So basically, phylostratigraphic analysis means that we associate to every gene's an evolutionary age. And we look, so in the differentially expressed genes, is we have novel representation of the most ancient genes or the youngest genes. And so you can go next. And this is what we found. So here what you have or these results of this analysis for two big aging, multi-tissue aging signature. And what we found is each time, so it's an important atavistic genetic expression in the most ancient genes, and also an underrepresentation of the youngest genes compared to the distribution of all the ages of human genes. So this, so yes, and we found this for this dissociation for skin senescent, mesenchymal senescent, immune ovarian, and human progenitor cells. So why it is dissociation? Because sometimes in the most ancient genes we have this overrepresentation, but sometimes it is more like downregulated genes that we have in the most ancient genes. So sometimes it's more upregulated genes. So it depends of the cell types, and we didn't find it for brain and stem cells that may be compatible also with differential aging data. And yes, that's it, I think. Yes. So at the end, the way we are seeing aging here is that during development, all the cells. So here, what you see is a cognitive icon. So during development, all the cells are aligned towards a big goal. So that is to reach the appropriate anatomy. So to do development. And once they reach development, they start to follow their own cellular goals that have a lower scale, and leading to misalignment and the gradual decline of the anatomy. So yes.
Great. So just to summarize, and then the slides will be done. There are three big ideas here that we're proposing. First, that a major aspect of the aging phenotypes are the problem of what does a cognitive system do after it has met its goal. And so basically you have cellular collectives which are very good at meeting certain anatomical goals, but when that's done, when the body has been constructed, the changes of maturation are completed, that they basically the collective starts to disband. The cells are no longer aligned in anatomical space. They pursue their own individual tiny little agendas. Part of that is the blurring of the bioelectrical patterns, the memories that keep the entire system aligned towards a species-specific shape. So in this case, a human anatomy. And of course, there might be, as Leo said, some sort of feedback loops that are kind of positive feedback cycles. And we can literally see this happen. You can see the cells are dissociating and disaligning from each other because they begin to express transcriptional signals that are going backwards across the evolutionary tree. So they are no longer, all of the cells of the body in an aging body are no longer on the same page about what kinds of genes in terms of the evolutionary phylogenetic tree, where what kinds of genes should they be expressing. And so the final thing I want to leave you with is this notion. And so Leo and I just wrote a chapter on this, talking about aging and longevity beyond the level of a single individual. So we really think that aging needs to be thought about more than how do we maintain a current standard human for as long as possible? Because if you take the long view with all of the technologies that are coming online, which are control over morphogenesis and bioengineering and so on, I think we think that the chances that a visitor that comes back to Earth even 50, I think, years from now, that he finds exactly the same Homo sapiens with the same body structure, the same susceptibilities to degenerative disease and astigmatism and all of these things, the chances that he'll find the same exact humans, I think, are very low. We are going to change. We are going to change both biologically and technologically in many different kinds of ways. We can talk about that. And the question of longevity isn't how long can you maintain in exactly the same body that's been here for hundreds of thousands of years, but actually what is it that we want to maintain far into the future? As we think about radical longevity, not decades, but centuries and beyond, the question is, how do you want to persist? And what do you want to be? Because it doesn't have to be the same thing that eons of cosmic rays and other kinds of mutations have left us with. So it's possible that we figure out a way to radically prolong a normal human body and give the cells the same goals over and over again. Maybe that's true, or maybe they actually will require considerable novelty in refreshing that goal state. And maybe, like the caterpillar, you can't really persist in exactly the form that you're in now for extended periods of time. But if you're willing to change and become something else, maybe you can. So that's something that remains to be seen. So at this point, we just want to thank the people who did a lot of the work that we mentioned today. So here are some postdocs and PhD students and our computational and aging groups and our funders who support us in this work. And Jeremy Gay drew the pictures.
That was great. Thanks, guys. So I have so many questions. And what's interesting is even though that was, 30-minute presentation, I feel like you guys could probably go for hours on so many segments of that, which we didn't have time to dig into today. So my first question/comment would be related to the anatomical compiler, which I've heard you talk about many times as you see this as we need to learn to communicate with our cells to basically give them that goal directedness, right? So what are your thoughts on, so it's funny because you thought that my focus is completely on diet, but in a sense, in a rudimentary way, I'm trying to not only learn, but communicate to what does my kidney want or my liver and so I'm looking at correlations in my own data, whether it's diet, supplements, et cetera, with biomarkers and trying to follow the pattern so that I can give the kidney what it wants so that it can stay as youthful as possible for as long as possible. In a sense, discovering, the signals, which I don't know I don't know what the mechanism is, but I know what biomarkers of youth look like. So the question basically is how do you foresee, the anatomical compiler, digging deeper than just tracking your own data and looking at correlations within one's own data, which is basically precision health or precision medicine, precision nutrition. How do you foresee us being able to, because you can't do patch clamp, and look at ion channel measurements. I don't know how you would do that in a big human, you can do it in cells, but right. So how do you foresee it translating the anatomical compiler into people?
Yeah, a few things. First of all, and I have to do an invention disclosure here, which is that there's a company called Morphoceuticals, which is spun out of our group, which is developing some of these technologies. Our goal absolutely is to get good bioelectrical signaling in large-scale organisms such as people, and there's technologies that we hope are going to be developed soon to allow us to do that in the long run. The point of the anatomical compiler is that it's a communications device. And one of the things that we're doing is using tools of AI and VR and some other things to try to develop ways to literally communicate with your organs. So when you talk about finding out what the liver and the other organs want, I think that's actually doable. Right? That's literally doable because these things are not passive machines. These are actually problem-solving agents that are continuously navigating these complex physiological state spaces and so on. And they do have goal states. They do feel stress when they're deviated from their goal states. And these are all things that we should be able to communicate with them about. If we can understand and basically crack the code and communicate with them on their own terms through various, the bioelectrical interface is the most obvious one. There are obviously others, biomechanical, optical, and so on. And so this has to start in model systems first. But I firmly believe that the fundamentals of what we're doing are extremely well conserved evolutionarily. And when we figure out how to talk to planarian cells, frog cells, mouse cells, we will have the tools that we need to figure out what aspects of the novelty of the goal states are needed for human cells to do what planarian cells are clearly telling us is doable. Right? We have this example that tells us that it is not a physical impossibility for an organism that's fairly complex. Can learn, they have a centralized brain and so on, highly regenerative, cancer resistant, and basically immortal. We know it's possible. So that's Leo. Anything you want to add to that?
No. You said a lot. No, I think at the end the question is where are the goals encoded and how we can rewrite them. And yes, here we have shown in the lab that it can work with bioelectricity in planaria. But now we need probably more data that we still don't have for humans, for example.
So would this involve like imaging, where you're going to image the whole human and then you're able to detect, the membrane potential again across the various organs. I mean, so you have a diverse range of membrane potential across different cell and tissue types, right? So how would you do that across so many different tissue? Like, what technology would you use to do that in people?
Yeah, so right now, the voltage sensing technology is optical, and so there are dyes that are being developed to do this. We are hoping to develop similar kinds of reagents that are not optical, but let's say CT, MRI, ultrasound, so that you can go into deep tissues and get voltage maps. But there's something important here. This is it's very likely that what you don't need for this is a very high resolution map of what every cell is doing, because that's not how the code works. The code is not a single cell code. This is a coarse grained measurement that is where the large scale patterns are what's important. And this is what we've been doing for about 25 years now, is trying to understand what is it about the bioelectrical states that the other cells are interpreting. And we know that it's not necessarily this very high level, high resolution information. It's large scale measurements. And yeah, and so we have another spinoff called Astonishing Labs, which is looking at various aspects of aging in general. And we have some amazing models, so we can talk about anthrobots and what they're telling us about aging and all of these kinds of things. The model systems first, but all of this I think is going to be applicable to humans.
Yeah, so it's the ion channel drugs and then imaging in the whole body human, right? So in order to get enough information, I mean, so if you use the ion channel drugs and then use whole body imaging, would you have enough data from one time point in order to say, well, this is the bioelectric state of the kidney, the bioelectric state of all the different tissues, or would you need multiple testing over time under different physiological states? I mean, because we assume that, say the fasted state at 8 in the morning, you'll get a certain bioelectric condition, but the bioelectric condition for various organs and tissues, even cells, stem cells, whatever, may be different, at noon, at 6 a.m., or after eating or after a workout, right? So would it have to be multiple time points under different conditions over a few month period, or how would you foresee that? How would you foresee really mapping the bioelectric state of the whole human?
Yeah, so this is the kind of thing we're figuring out now in these model systems. The signals, there are going to be signals at multiple timescales. So as you said, there are going to be metabolic signals that are part of the bioelectric readout that are not what we're looking for. And there are going to be very slow changing long-term kinds of gradients that are basically saying, this is what your kidney should be shaped like. This is what your liver should be shaped like when the liver cells, die and need to be replaced. Here's where the new cells need to go to maintain a human liver. Right? Those are the kinds of signals we need. And that's the thing. I will also say that we had an interesting. We have an example where we were able to successfully sharpen these kind of degraded voltage gradients, and we did that in this case of birth defects. So we found that there are certain kinds of teratogens that basically make these voltage gradients blurry, and they cause birth defects. We were able to go in and we had a very nice bioelectric picture of the brain and of the early brain, and we were able to sharpen that up and get normal brain development in the context of some really nasty genetic mutations, teratogens, and so on. After that, we wanted to do the same thing to fix disorders of the gut and heart. But we didn't have voltage imaging of the gut and heart. They were deep inside the embryo, and we just didn't have the maps. It turns out that we did exactly the same thing that we did for the brain, and it ended up repairing heart, gut, and face phenotypes, even though we never had good voltage maps for that. So I think part of this is going to be that there are probably well-conserved general themes that we can capitalize upon without having necessarily all of the information at once. But we won't know that until we do a complete mapping in some model system, and then the human. What do you think, Leo?
Yeah, this is an empirical research program. We still don't know exactly, but it's once to answer your question, it's always better, I think, to have more time points. Because we will know exactly what to do. Yes, yeah. Yes, yeah. That's mainly biological data that we need in model organisms.
And the challenge here is it seems like your lab, granted as big as it is, I mean, I don't know of anybody else using this approach in terms of with the mindset of aging, cancer, rejuvenation. So, it's like how long of a time scale? I hate to put the classic, are we 5 to 10 years away? I know we're not. I know it's going to take a lot longer than that. But, I don't want this to be where 100 years from now, people will say, oh, well, Michael Levin's group laid the pathway, for all of us to have this now 100 years later because there were just so few labs. So, what kind of timeline do you foresee this happening?
Okay, clearly I can't tell you exact timelines. It depends on how the science goes. It depends on the funding, regulatory. It depends on a million things. However, I don't think it's going to take 100 years. I think I think we're going to get to see this stuff. And just to be clear, there are a number of other excellent labs doing bioelectrics research. So people like Min Zhao and Emily Bates and lots of other people that are doing bioelectrics. And there are people certainly looking at regeneration and its relationship to aging, which I think fundamentally the solution to aging is going to be to activate regenerative responses. Where that's the big sort of roadmap. There are other labs looking at this stuff, and I think it's all going to converge. And I think there are both technologies and conceptual frameworks. Actually, I think the biggest thing holding it back is this real phobia that people have, especially people schooled in molecular and cell biology, of using the tools of cognitive science to understand what their system is doing. People tend to, outside of the diverse intelligence community, people think it's really weird when we talk about cell learning and goal states and motivating cells and wiping priors and all these kinds of things. I think this is actually the biggest blocker. But in the technology sort of close after that. But these things the ice is melting. These things are, the stuff that we've published in, Cell and Trends in Genetics and whatever. We can now talk about these things, which was basically impossible just a few years ago. It's going to happen, and I don't think it's going to take 100 years.
Nice. The argument against that is, as you say, often people are bad at recognizing unconventional intelligences. And in the case, the unconventional intelligence in the applied application of bioelectrical, restoring bioelectrical, why can't I talk? Restoring bioelectrical patterns is you guys, right? So, and slowing down the process, right? It's not the cells that are the unconventional intelligence and what they're doing. It's you guys driving the ship. So hang on, wait, just besides that comment, there's so many rabbit holes to go down here. So as you mentioned, restoring the bioelectrical gradients in the brain, whether it comes to birth defects or otherwise, I mean, is that the biggest fish? I mean, just, you could make the argument that, okay, within cells and the tight, the tight junctions between cells and the signals to cells, right? That decays over time. So you get the blurry cell gradients and all that. But the signal coming from the brain, or signals coming from the brain to the cells, obviously too, the brain has its own issues with the bioelectrical patterns probably decaying over time. So is that the big, billion-dollar question? If you fix the bioelectrical patterns in the brain, everything else follows.
Well, I think you're talking about two related questions. One is, to what extent are signals from the brain actually important for managing the rest of the body? There's some evidence for that. I mean, there's certainly a role of the nervous system in limb regeneration. We've shown that there's a couple of aspects of development that don't go quite right when you don't have a brain. But by and large, the brain, you don't need the brain to do most of morphogenesis, right? Like an incredible amount of morphogenesis happens without a brain, before the brain forms. It's not at all clear that you need the brain for the kind of upkeep that we're talking about. The other thing you're talking about is the maintenance of the brain itself. And I think, and I'm sure Leo has some thoughts about this too, but I think it's a very interesting question as to whether the kinds of cognitive changes that we see in aging are a hardware problem or a software problem. That is it that if we were to maintain if we were to continuously rejuvenate the cells, would we all have the plasticity of a 20-year-old? Or is after some number of years, maybe it's 100 years, maybe it's 1,000 years, but after some number of years, a cognitive architecture like a human just gets stuck in its ways in some ways, right? We don't know. That's a major open question. What do you think, Leo?
Well, I don't remember exactly the number, but we wrote in the last paper. So it seems that for some speculative number that we have 300 years of memory in our brain of video streaming, let's say. So we don't know exactly what will happen if we live like for a very long time. So this is a real question if it is just a hardware or software problem that is happening during aging, or if it is just an adaptation also to aging. Um, but yes, at the end it will be, yeah, as Mike said, the brain control lots of things, but we can even do, I think, morphogenesis without the brain now for some model organisms. And here we are more talking about the general collective intelligence, so of the body. And then if we can reactivate the developmental program, the regeneration, it will be the equivalent to rejuvenation.
So I guess the argument against the brain being an important, key component in this process is the work you guys have done on planaria, where you cut the head off. And actually, I think it's something like 200 something parts. You cut it into some 200 something parts, and each of them will grow into a new planaria and regrow a brain that has memories that have been encoded somatically, right, on the body. So I guess that's the argument why the brain is not so important. But how does that, does that suggest that, people who have Alzheimer's disease, if you could figure out how to get the cells in the body that clearly don't have amyloid and, amyloid plaque buildup, can you get the cells in the body to then retrain the brain to have the right bioelectric networks to basically reverse Alzheimer's? Is that how you would read that, or is there a different way to interpret that?
So we don't know yet if mammals and humans in particular are going to do this amazing trick that planaria do, which is to regenerate their memories along with their brains. We don't know the answer to that. But my suspicion is that the standard story of how, I mean, nobody knows how memories are actually stored in the brain, right? Like we don't actually know. But the standard story around synaptic structures and things like that, I think is not right. And there are a number of other people now that are sort of studying alternatives to that story. But I think one of the most interesting clinical data sets that speak to this are the rare human patients with extremely reduced brain volume and normal IQs. So Karina Kaufman and I did a review of these cases recently where there are some people with just like amazingly little brain tissue and overtly normal IQs, normal personalities, and so on. So the mapping between cognitive performance and the sort of neural real estate is not obvious. It's not as easy as as clear as we think it is. And where these memories are, I mean, we like to think they must be at least in planaria in the rest of the body. I'm not sure that's true. We can get into that. That would be a whole other discussion. But until we really understand how that kind of information is stored in the brain to begin with, we can't have any strong opinions about any of this. Just the fact that general anesthesia works, right? So if somebody had come to me and said, okay, I got a reagent, what it's going to do is disconnect, as general anesthesia does. It's going to disconnect all of the electrical synapses in your brain, but don't worry, eventually it'll sort of wash out, and then everything will go back to its correct electrical state. I would say never in a million years, right? Like yeah, they'll go back, and you'll end up somewhere, and it will never be anything. Just the fact that anybody ever not everybody does, but most people wake up the same eventually after general anesthesia, that already tells us that there are certain kinds of stability that we don't really understand very well.
Yeah, so many rabbit holes to go down. So the first one would be for the people who have a minimal brain volume, with the hypothesis, or part of the hypothesis be that they're doing compute systemically somehow, and then that translates to the minimum amount of brain volume that they have. So that's how they're able to have a somewhat normal IQ, because they're doing the compute everywhere else as much as possible.
This is to really do this justice, we'd have to have a whole other conversation about the closure of physicalism and some wild stuff like that. The one thing that I do think we can say is that the standard arguments about redundancy and so on, look, we are under such metabolic pressures, right? There's a lot of pressure in terms of childbirth to not have a giant head, right? And if you didn't need to have all this brain volume, you could kind of do it, and it wouldn't be this big. There's a lot of pressure against that, and the same thing for for the rest of the body. If you could do all that compute in the rest of the body, you probably would be already. I think actually the explanation is much weirder than that, but that'll be a different discussion, I think.
Not to push it into the realm of sci-fi, but the classic gray alien has it's a giant head. It's not tiny head, right? So having the compute in the brain's got to have some, if that's a real thing, right? If it's got to have some evolutionary advantage.
I don't know. Maybe they design their heads. They're not born that way. I don't know what the gray aliens are doing. So,
all right. So planaria, right? So that they're immortal and that they, so all right, wait, goal directedness and planaria. So how, I think you've mentioned in a couple talks where they kind of get bored, but then they reset it after a period of time and then they're back on track. So it's not like a linear process where they're constantly regenerating. It's like, that maybe it's biphasic where it's, but they always get back on track, right? So how is it that planaria have that, I guess it's evolutionary where they evolved it, but that sense of goal directedness where they're always going to regenerate, always going to rejuvenate, no matter how many parts you cut them into. And then how can we use that same, if it's bioelectricity, how can we do that in people?
Leo, do you want to say anything about worms, and then I'll talk about the evolutionary aspects of it?
Yeah, I was just thinking. This is a good question. They probably have never-ending anatomical goal that is here, constant regeneration. It just maybe takes time to do it sometimes. It depends on the window you are looking at it. But I think they have, and it explains why they just don't age. Well, at least this is the prediction of the simulation we have and and the way we are seeing the loss of goal directedness. We are seeing aging as a loss of goal directedness. But yes, the idea would be probably to do the same one day in mammals. The question is how we can do that. So because what we in the framework of the loss of goal directedness, what happens is just after development we don't have any anatomical goal anymore. So we need to rewrite it. So, but there are different kind of goals. Probably there are the goals of maintenance. So there are goals of total regeneration. But maybe we need, as Michael introduced, said it, we maybe need more novelty. So maybe we need to define a complete new goal state in the anatomical space. This is still an open question here.
Yeah, I mean, I think what the planaria are doing is refreshing the. I mean, every couple of weeks they tear themselves in half, right? And they give themselves this new challenge of regenerating, and it's entirely possible that as humans, if we were to crank up that regenerative capacity, that we would be able to refresh these goals ourselves. The reason I think the reason that planaria have it natively, and I do think we can induce this in humans, and I think it's possible, but the reason planaria have it natively, I think is quite interesting. It bugged me for decades. It bugged me that the animal with the best regenerative capacity, cancer resistance, immortality is the one with the noisiest genome. And that's because these guys reproduce by tearing themselves in half and regenerating. So they keep all the mutations. They don't throw away somatic mutations like we do every generation, where only the egg gets, and the sperm gets going, and the rest of the body and its mutations are lost. They have an incredibly noisy. I mean, they can be mixoploid. Every cell could have a different number of chromosomes, right? It's crazy, and yet they're the ones with the most anatomical fidelity, right? What classes have you ever had in biology where they would tell you that the animal with those properties should have, in fact, not the tightest genome, but the noisiest one, right? And so I think what happens in planaria, and we've modeled this in a couple of papers. We've simulated this, and you can see exactly what happens when the material is really unreliable. When evolution is dealing with an extremely unreliable substrate, right, where you don't know what your genetics are going to be. Every cell might be different. You don't know how many cells you have. You can't count on the hardware. What you can do over time, and we see evolution doing this, is focusing on improving the algorithm. It improves the communication between cells such that it becomes incredibly tolerant to problems that are happening in the hardware. There's a whole spectrum of these, right? So something like a C. elegans is probably way on the left of that. Every cell is numbered. They don't really have a lot of plasticity. And then you have mammals, and then amphibians, and then planaria are sort of all the way on the right in terms of how much tolerance do you have for getting back to where you need to be anatomically from various weird starting positions, not only in terms of injury and when the environment does something to you, but your own internal components are you get mutated. That's an injury to the internal information. And so I think there are different degrees to which cell collectives are good at improvising solutions in novel scenarios, and it goes all the way back. It's sort of built in, but some species naturally take it to a very high level. We're going to have to crank that up, but I think that is possible.
And a key component of that equation is the ability of a planaria to compute, right? So the barium experiment that you've mentioned many times, where you put a planaria in a barium and their heads explode, but then if you give them enough time, they figure it out. And this isn't, this is like adaptive evolution within the same organism, and not over generations within the same planaria. So, but that is inherent in the ability to compute. So you could make the argument, I guess, that planaria have evolved to be better at computation and solving problems than human cells?
I wouldn't make that argument. I think all cells do it. We don't know. Okay, there may be differences because very little of this has been studied. But look, when we make tadpoles, so Doug Blackiston in my lab made tadpoles where the eyes are on their tails. And when you have a tadpole with only the eyes on the tail, out of the box, they can use those eyes to do visual behaviors. They learn in visual assays. Why does it not take generations of mutation, selection, evolution? You have an animal with a radically different sensory motor architecture. Those eyes don't even connect to the brain. They connect sometimes to the spinal cord, sometimes to the gut, sometimes nowhere at all. And those animals can see. Why does that work? Why does that work out of the box? And I think specifically because that ability of evolution doesn't make solutions to specific environments, it makes problem-solving systems. And the reason those tadpoles work is because the tadpole could never count on having the eyes be in the right place to begin with. When they are, great, and you get the default outcome. But when they're not, it's the same problem solving that every embryo has to do. And I don't think it's specific to planaria, although there may be different skills and different cell types, so that remains to be seen.
So was it in that experiment where it was the bioelectric patterns were altered such that they grew the eye in a different place, so they didn't even need to have the genetics for having that second eye, but it was just you guys hit it with a different bioelectric pattern, and then you were able to teach them to grow a second eye?
There are two. So there are two ways to do it. One way is to do it bioelectrically, and you can do it that way, and then you don't touch the genetics. The other way, you also don't touch the genetics. What Doug actually did was to take eye primordial cells and just implant them, just stick a bunch of them into the tail, and they built an eye. So that's another way of doing it.
Yeah. All right. So rather than that approach, the first approach, using the bioelectricity to basically guide development to regrow another eye, sure, or in any location on the body that you would want, is that the future of human regeneration, right? So as you showed in that slide where you've got epigenetic reprogramming, the bioelectrics are upstream. So if you hit it with the right bioelectric signal, you've got epigenetic reprogramming, you've got potentially extension of telomeres and all of those other aging mechanisms. So this kind of ties back. I know you guys got to run, but what's your pitch for why bioelectricity is going to basically drive all of the stuff downstream, whereas everybody else is thinking about CRISPR and all of the, bottom up rather than the top down?
Well, there's two questions there. One is there a top down approach at all? And the second question is, if there is the top level bioelectrical or something else, right? So here's what we know. We've demonstrated, and other people have too, in other ways, lots of examples where you give an incredibly simple high level intervention, right? So in the leg regeneration work, we in 24 hours of stimulation with something as simple as monensin. It's a sodium ionophore. It's not patterned. You don't turn it on and off. We can't crank it up or down. We just soak the animal in monensin 24 hours. A year and a half of leg growth. We don't touch it after that. We don't tell it how to grow a leg. By the way, the same monensin induces a leg in an adult frog. It induces a tail in a tadpole with an amputated tail. Same monensin. The cells know what goes there. It's a high level trigger. When we induce the eyes, I don't know how to tell it to do all the different patterning of the lens, the retina, the optic nerve. We have no idea how to do that. What we do know is how to say, build an eye here. right? And all of the other stuff happens. There are many examples like this. So we already know top-down programming works. Is it going to be bioelectric? I think so. We've shown that with very simple bioelectrical patterns, all the biochemistry is downstream. The signaling factors, the gene expression, the cell movements, the epigenetics, all that stuff gets in line. Might there be something upstream of that? Possibly. We haven't seen that yet. As far as I can tell, it's bioelectrics, but the future will show. Anything am I leaving out, Leo?
No, I think the future of medicine will be in the top-down control. And if we come back also to your previous question on planaria, is genetically it's horrible. They have myxopoid cells. They accumulate genetic mutations. So in a sense, mammalian cells are more genetically damage resistant, but still they don't regenerate. So everything is the algorithm, the software of life. And I think here we come back to the top-down programming. So yes, if it is biological, maybe, yes, we have a big bias, of course, maybe something else, but yes. So
Nice.
Yep. I'll just give you a very brief story about what else could possibly be upstream of that. And it's the story of this guy named Albert Mason, who discovered the field of hypnodermatology. So back in the 50s in the UK, he was delivering babies, and he didn't like using anesthesia because it suppressed the breathing of the babies. And so he decided to try hypnosis, and he was successful at it. He was delivering babies with hypnotic anesthesia. And I guess some of his colleagues were sort of making fun of him, and they said, "Well, here, you think hypnosis is so good? Try this." And they gave him a case where there's a young teenager who had ichthyosis of both arms. So his the skin is basically black, it's cracking, and just unbelievable. The picture is, I think it was the British Medical Journal, if I remember correctly. The picture is there, it's just incredible. So he says, "All right, what the hell? Well, I'll try it." And he gives the kid a suggestion that one of the arms is gonna clear up. And care to guess what happened, right? And that picture is in the paper. And so he establishes the field of hypnodermatology. So he has a practice. People come to him with all sorts of skin conditions, and he sort of goes top down, and he talks to them, and he talks to the skin cells, and he has good effects. So he does this for some number of years, but that's not the end of the story. The end of the story is more interesting. He does this for some number of years, and he discovers that their skin diseases clear up all right, but they start to have problems in other areas of their life. They start to gamble. They start to, have marriage problems. They do weird, behavioral things. And what he realized was that as he said, and there's 4 interviews with him that are amazing that you can listen to online. What he said is he realized that he went one level above the drugs that everybody else was trying to use in talking to the skin cells. He certainly got up a level, but at the level that he was at, he was still only shoving the pile somewhere else under the rug. He wasn't really resolving it. And so he went into, I guess, psychiatry and being a psychiatrist and trying to resolve the sort of cognitive issues that people have that presumably can work out through somatic symptoms and so on. So, the question becomes, in operating at this bioelectrical level, are we actually hitting the very highest level? Possibly not. And I do think this is why in my group, all of this stuff is very tightly integrated with very fundamental questions of cognition and memories and goal states and so on, because I in the end, I don't think it's the physics of bioelectricity that's important. It's the mechanism of bioelectricity playing out the basal cognition of cellular collectives. And so maybe there's something else that we can trigger at that point.
And consciousness, and then where is consciousness? Where is platonic space? How can we get there? How can we find that? So I think you guys are all hard out. I don't want to keep you. I have so many other questions: anthrobots and xenobots, and metabolomic studies on rejuvenation and axolotl and deer antler and human stem cells. But hopefully we can save that for another day. I can get you guys back. So
absolutely, yeah. Let's talk again.
All right. Ciao, guys. Appreciate
Thanks very much, Michael. Yeah.
Thank you. Thank you.
See you, Leo.
Bye.
See you. Bye.